In 2002, a woman in Washington state named Lydia Fairchild applied for public assistance while pregnant with her third child. As part of the process, the state ran a routine DNA test to confirm she was the mother of her two existing children. The results said she wasn't. Not a partial match, not an ambiguous result, a flat exclusion. According to the DNA, Lydia Fairchild was not related to the children she had given birth to, breastfed, and raised. The state accused her of fraud. Prosecutors moved to take her children away, and the reason had nothing to do with lies, mistaken identity, or a lab error. Lydia Fairchild was a human chimera, a single person built from the merged cells of what should have been fraternal twins. Fraternal twins normally start as two separate fertilized eggs, developing side by side in the womb as two distinct individuals. But every so often, in the earliest days after fertilization, before either embryo has developed into anything resembling a person, those two embryos fuse together into one. Instead of becoming twins, they become a single individual carrying two separate sets of DNA. One lineage in some tissues, a different lineage in others. Scientists call this Tetragametic chimerism because it takes 4 gametes, 2 eggs and 2 sperm to create what ends up being one body. The condition is exceptionally rare. Fewer than 100 cases have been confirmed in medical literature since it was first identified in 1953. And because most chimeras never get tested in the way that would reveal it, many carry 2 sets of DNA their entire lives without ever knowing it. A chimera's blood might carry one genetic profile while their skin, hair, or reproductive organs carry another entirely. In Lydia Fairchild's case, the DNA in her blood, skin, and hair did not match her children at all. The lineage that matched was hiding somewhere else in her body. Most chimeras look completely ordinary, but not all of them. Some show visible signs of carrying 2 cell lines at once. Patchy skin pigmentation is one of the more common markers, in areas of noticeably lighter or darker skin that don't match any typical birthmark pattern, sometimes covering as much as half of the body. Heterochromia, having 2 different colored eyes, shows up in some cases too. In rare instances, a chimera can be born with ambiguous genitalia or a mix of male and female reproductive anatomy, because the 2 fused cell lines were carrying different chromosomal instructions for sex development. One American singer discovered her own chimerism in 2009 after investigating a birthmark that ran straight down the center of her body, a visible line where her 2 cell lines seemed to meet. Blood tests can reveal chimerism too, even without any visible signs. A chimera can carry 2 entirely different blood types at once. In one documented case, a woman's blood tested at 61% type O and 39% type A, a mixture that only makes sense if 2 separate genetic lineages are both actively producing red blood cells inside the same body. Lydia's case might have ended in tragedy if not for a similar one that came before it. In 2002, the New England Journal of Medicine published details on a Boston woman named Karen Keegan who needed a kidney transplant and, as part of the screening process, had her family tested for tissue compatibility. The results showed that 2 of her 3 adult sons were not, genetically speaking, her children either. Doctors eventually determined that Keegan carried 2 distinct populations of blood cells and traced the mismatch to tetragametic chimerism. Lydia's attorney, Alan Tyndall, came across the Keegan case while searching for any possible explanation for his own client's impossible test results. It gave him a theory to test. If Lydia was a chimera like Keegan, then the matching DNA might exist somewhere in her body, rather than her blood or skin. Doctors ordered a cervical smear. That sample, taken from the tissue closer to where her eggs actually originate, finally matched her children's DNA. Lydia Fairchild had been telling the truth the entire time. The charges were dropped and she kept custody of all 3 of her children. That were her children, but that would be incredibly scary. Chimerism doesn't just complicate maternity cases. It has done the same thing to fathers. In one documented case, a man conceived a child through intrauterine insemination, and a standard paternity test excluded him entirely. But a follow-up ancestry test revealed something stranger. The relationship between the man and the child looked less like father and son and more like uncle and nephew. Researchers eventually confirmed the man was a tetragametic chimera. His unborn twin cell had ended up inside his own reproductive organs, meaning the sperm that conceived his son actually carried his vanished twin's DNA rather than his own son. Genetically, in a strange technical sense, his absorbed twin was the biological father. Cases like this definitely matter beyond the shock value. In the United States alone, more than 380,000 legal paternity tests are ordered every year for child support, custody, and immigration cases. Standard testing usually relies on a single tissue sample, blood or cheek swab, and has no way of flagging a chimeric result unless someone already suspects this condition. Geneticists who have studied these cases believe chimerism is likely more common than the confirmed case count suggests, simply because most people are never tested in a way that would reveal it. Chimerism isn't unique to people. It also shows up in the animal kingdom too, most visibly in male tortoiseshell cats. Tortoiseshell coloring normally only appears in female cats because it depends on having two X chromosomes, each carrying instructions for a different fur color. A male cat with tortoiseshell patterning got there because two embryos, each carrying different colored genes, fused together early in development, giving him patches of both colors and, effectively, two sets of feline DNA in one body. These cats can occasionally survive to adulthood, although their extra genetic material involved usually leaves them infertile. Stanford geneticist Barry Starr, who worked on one of the paternity cases, has pointed out that chimerism barely registered as a research topic until DNA testing became widespread and routine. As genetic testing keeps getting cheaper and more common, more of these hidden cases are likely to surface. Not because chimerism itself is becoming more common, but because we finally have a reason to go looking for it. Every so often, a case like this becomes public because a DNA test forced the question. But geneticists suspect the real number of chimeras walking around is far higher than roughly 100 confirmed cases on record. Most people never have a reason to test more than one part of their body, so a second hidden set of DNA could sit undiscovered for an entire lifetime. Lydia's case became a legal landmark specifically because someone finally thought to look somewhere else. Most chimeras never find out. They go through life with two sets of DNA quietly coexisting in different parts of their body, with no symptoms and no reason to ever get tested. Lydia Fairchild only found out because a state agency demanded proof of something that should have never needed proving in the first place, her parenthood to her own children. But her own body, in a very literal sense, had absorbed evidence of a sibling she never met, a twin who never got the chance to become a separate person, and instead became part of her. She had been right about her own children the entire time. The only thing wrong was the test. Thanks for watching, and I'll see you in the next one.